Now, one type of microchromatography technology that has been on the market for quite a few years are pipette tip columns, where people pack resins into the end of pipette tips. And there have been several versions of this introduced. And one question that we get is how is AssayMAP different from the pipette tip column? In both the probe syringe and the syringe form and the spin format mode, there are separate entry and exit ports. The liquid either enters in the top, it goes out the bottom, or vice versa in both of these modes. And so we can actually pass liquid through the column in a single pass and have it go out the other side. And this can be done in both modes at a reasonably controlled flow rate, very precisely controlled with the probe syringe and reasonably well controlled in the spin format. And both of these systems have been very carefully designed to prevent air entrainment or to be able to very reliably remove air from the bed if it does happen to get in there. Now, in a pipette tip column, you're actually using air displacement to move the liquid in and out of through the bed. And there's only one entry and exit port at the distal end of the pipette tip. So the only way you can get contact between the liquid and the bed is to pull it in and then push it back out again. And it's basically like sloshing the liquid back and forth through the resin. So this is basically an equilibrium absorption type of process, whereas in the AssayMAP system we can actually do true flow-through chromatography. Also in the pipette tip column, because you're relying entirely on air displacement, the flow rate is highly variable, and it varies during the whole time that you're running it. And it actually, in order to overcome surface tension, you have to run at a relatively high flow rate in this system. In addition, because it's an open system and you're using air displacement, air entrainment is actually almost a certainty. You're almost certain to get bubbles in there, which contribute to the lack of quantitative binding and elution. So if we look at how the system works during the loading, washing, and elution phase, the major differences in washing and elution. In AssayMAP, we can do true chromatography, a single pass of wash buffer or elution buffer through the packed bed will very efficiently wash or elute from the bed in something like 5 to 10 column volumes, kind of typically what you would expect to see on a chromatography system. In the pipette tip column, you're working in this equilibrium absorption mode, so at the end of a cycle of multi-pass equilibrium washing or elution, the liquid in the well at the end of that is the same as the liquid in the packed bed. And so in order to get complete washing or complete elution, you have to go into a fresh well and do it again in order to pull out that last little bit, and you never get everything. And so, number one, you're diluting the product more than you normally would, and it's very, very difficult, if not impossible, to get quantitative results with this type of system. So, on to applications. What can you do with a high-throughput microchromatography platform? Most of the applications involve purification. So, you equilibrate the column, you selectively bind something from the sample, you wash away the non-bound materials, and then elute the product, collect it in a microplate well, and then either read it in a plate reader or you have the purified material available for subsequent analysis by something like mass spec. And this is an example of using protein A to purify monoclonal antibodies from various types of feed streams. And this is extremely quantitative in the AssayMAP system. On the left, you see some work done in the spin format. We've plotted the A280, just the protein absorbance measured in the microplate, as a function of the mass of antibody loaded. And you can see that it's very linear up to a range of about 100 micrograms load, at which point it begins flattening out because the column is beginning to reach its saturation binding capacity. What we've done here is do 3 different sample volumes. So there's actually a very broad range of concentrations measured here. But because we've plotted it on a mass basis, you can see that all the curves basically overlap. That's a really good indication that we're getting perfectly quantitative binding elution. On the right are some work done on the Bravo system with the probe syringe technology. And these are 3 standard curves with 50 microliter samples in the linear range. And you can see exactly how linear it is. And there are actually 3 curves overlapping there. One are standards and buffer, and then 5 and 10 with 5 and 10 mgs per mil background protein, which is actually nasty fish skeleton material that's used as a very sticky blocking reagent in ELISA assays. So it's kind of a worst case scenario. And you can see we're getting very quantitative purification out of that nasty sample matrix. Here's a real-world application, some work done by Genentech. This was also done in the spin format, but run on a TCAN automated system. So this is all fully automated. And these are actual cell culture supernatant samples from a process development laboratory, comparing the AssayMAP results to the gold standard method, which is protein A affinity HPLC. And you can see there's a very good correlation between the two. The numbers that you get in a direct comparison are virtually identical, and the analytical precision is very high for both methods. The difference is the AssayMAP method has somewhere between 50 and 100 times the sample throughput of the HPLC, because you're able to run 96 cartridges or 192 cartridges at a time, as opposed to 1 at a time on the HPLC. Now, protein A is, of course, not the only thing that you can use as an affinity binding reagent. There are other protein affinity agents that you can use, and most people work with either antigens or antibodies as selective binding reagents. So we've created what we call the immunoaffinity toolbox, which is a relatively small number of surfaces, some additional specific protein reagents for particular applications, but also 2 products, a hydrophobic binding surface and a streptavidin binding surface that allows you to couple biotinylated reagents. And these can be used in a whole range of different applications where you're either targeting antibodies, either generally or class specificity or by their antigen binding characteristics, or you're turning around and using the antibody itself as the affinity ligand and capturing non-antibody antigen molecules as the analytical target. Now here's an example. This is using one of the capture select ligand, which is a small molecular weight protein ligand that's actually an isolated binding domain from camelid antibodies that's manufactured on a large scale in yeast. This is a technology developed by BAC in the Netherlands. And they make these on a custom basis, but they also have a standard range of ligands for a range of different applications in the antibody world, and also plasma proteins and biosimilars and some other types of applications. What we're looking at here, though, is an application that's very common in preclinical pharmacokinetics, where you would inject an animal with a drug, in this case an antibody, and then take blood samples periodically and monitor what happens to that antibody in the drug, in the animal. And you need to be able to pull out the antibody and purify it in order to both quantitate it and to see what happens to it. Protein A is not suitable for this because protein A not only binds the human antibody that's your drug, but it also binds the rat antibody that's normally in the rat. And you can see that on these gels. These are 20 microgram and downward serial dilutions. On the left is in buffer, so you can see the protein A does purify it out, but as soon as you put it in rat serum, the rat IgG just totally overwhelms it. On the capture select ligand, this is one of these ligands that's specific for human antibodies, but doesn't bind the rat antibodies at all. And you can see that the serum, or the buffer and the rat serum look virtually identical. So that means that we're able to selectively purify this antibody very effectively and very simply out of animal serum for PK studies.